Our today's topic on plant embryology, we study embryosac, the female gametophyte. Functional megaspore represents the first cell of the female gametophyte, as shown as the diagram. Mostly callagel and grows along the micropylar callager axis. The nucleus undergoes 3 mitotic division and from 8 nuclei, as shown in the diagram. These nuclei are reorganized. One nucleus from each group at a pole migrates to the center of the cell called polar nuclei. The 3 nuclei left at the callagel end are surrounded by walls and form antipodals. These 3 cells are known as antipodals. The 3 nuclei located at the micropylar and constitute the egg apparatus. One serves as a egg or female gamete and the other 2 as a synergids. The whole structure are 2 polar nuclei, 3 antipodals, 1 egg and 2 synergids. Is the mature female gametophyte or known as also embryosac. The embryosacs divided in 3 types: monosporic, bisporic embryosac, tetrasporic embryosac. The basis of classification: the number of megaspores taking part in the development of embryosac, the number of divisions occurring in the nucleus of the functional megaspore, organization of the nuclei in the mature embryosac. First one is monosporic embryosac. Monosporic embryosac develops from a single megaspore, and as such all the nuclei present in this type of embryosac are genetically similar. They are also classified by monosporic embryosac. First one is polygonum type, nucleate, most common type. First time described in polygonum, develops from the callagel megaspore. The nucleus is divided thrice to form 8 nuclei embryosac. In this picture diagram, we show the steps in development of polygonum type of embryosac. Second type is Oinothora type. 4 nucleated embryosac, egg apparatus 3 cell and polar nucleus single cell. Develops from micropylar megaspore, does not have antipodals. Example is Ongraceae family. Figures on the development of Oinothora type. Bisporic embryosac develops from one of the 2 dyads forms as a result of the first meiotic division of megaspore mother cell. Both the nuclei of the functional dyad take part in the formation of embryosac. Each nucleus undergoes 2 mitotic divisions and the mature embryosac is 8 nucleated. The 8 nuclei are organized into antipodals, egg apparatus and polar nuclei as in polygonum type of embryosac. The 4 nuclei derived from one megaspore nucleus are genetically different from the other 4 derived from the second megaspore nucleus. Figures shown the development of bisporic embryosac. The next type is tetrasporic embryosac. Meiotic division of the megaspore mother cell is not accompanied by the cytokinesis, and hence all the 4 haploid nuclei lie in a single cell called cynomegaspore. All 4 nuclei of cynomegaspore participate in the formation of embryosac, genetically more heterogeneous than bisporic type of embryosac. The tetrasporic embryosac are further divided on the basis of following criteria. The first one is the position of haploid nuclei in the cynomegaspore, the number of times these nuclei divide, organization of nuclei in the mature embryosac, types of tetrasporic embryosac. Divided in two categories, non-nuclear fusion occurs Adoxa, Plumbago, Pinia, Peperomia and Drusa type. After the second meiotic division, 3 megaspore nuclei fuse to form triploid nucleus. On the basis of these are divided in Fritillaria type and Plumbagella types. The first one is Adoxa type. It has 8 nuclei which are formed by the mitotic division of the 4 haploid nuclei of the cynomagaspore. The arrangement of the 8 nuclei in the embryo sac is the same as in polygonum type. Example for this Adoxa, Sambucus, Almas, Tulipa, Erythronium. Second is Plumbago type. This type of embryo sac is characterized by the absence of synergids and antipodal. This is the very important character. Out of 4 haploid cynomagaspore, 1 migrates to the micropylar end, 1 at callagel end and 2 at the lateral sides. Each nuclei divides again and formed 4 groups of 2 nuclei. One of the nucleus from each group moves to the center of the cell and form 4 polar nuclei. The remaining nucleus at the micropylar is cut off by a membrane and form the egg, on where there are no synergids. The other 3 nuclei usually disappear, but occasionally they too may be cut off by membranes. Example belongs to Plumbaginaceae family. Next type is Pinia type. The 4 haploid nuclei of cynomagaspore undergo 2 successive mitotic divisions, forming 16 nuclei. These nuclei arrange themselves in the 4 groups of 4 each, one at the micropylar end, one at callagel end, and one each on the 2 lateral sides. Now one nucleus from each group migrates to the center, and these 4 nuclei in the center form polar nuclei. The 3 nuclei at the micropylar end are cut off by membranes and form the egg apparatus. The remaining 3 groups of nuclei degenerate at maturity, highly polyploid condition. Primary endosperm nucleus is formed after double fertilization. Example belongs to family Paniaceae, Malpighiaceae and Euphorbiaceae. Peperomia type. The egg apparatus of Peperomia type is characterized by a single synergid. The 4 haploid nuclei of cynomagaspore undergo 2 successive mitotic divisions, forming 16 nuclei. 2 nuclei at the micropylar end form egg and a synergid. 8 fuse in the center of the cell to form a polar nucleus, and the remaining 6 at the callagel end form antipodals. Examples Peperomia and Ganera. Drusa type, 16 nucleate embryo sac. This type of embryo sac is characterized by a large number of antipodals. In the mature embryo sac, 3 nuclei from egg apparatus to act as a polar nuclei, and the remaining 11 nuclei are cut off by membrane and form antipodal cells. The number and organization of nuclei may vary due to irregularity in the divisions. Examples this Drusa, Rubia, Chrysanthemum, Almus, etc. Fritillaria type. The 4 haploid nuclei of the cynomagaspore arrange themselves in 2 groups, 3 at the callagel end and in the form of a triploid nucleus, and 1 haploid at the micropylar end. The triploid callagel as well as the haploid micropylar nucleus undergo 2 mitotic divisions, and as a result 4 triploid nuclei are formed at the callagel end and 4 haploid at the micropylar end. In mature embryo sac, 3 haploid nuclei organize into egg apparatus, 3 haploid into antipodal, and remaining 1 haploid and one triploid nuclei move to the center where they fuse to form a tetraploid polar nucleus. Example for this: Fritillaria, Lilium, Piper. Last one is Palmigilla type. The initial development is similar to Fritillaria type, and a triploid nucleus is formed at the calygeal end and a haploid at the microphylar end. Each of these nuclei undergoes a single mitotic division and form groups of two nuclei each. One triploid nuclei from calygeal end and one haploid nucleus from microphylar end fuse at the center and form tetraploid polar nucleus. One haploid nucleus at the microphylar end and forms the egg, and one triploid nucleus at the calygeal end, the single antipodal. There is no synergids also.